Good morning, fellow Spartans. Good morning. Thank you, guys. We're about to start, so thank you, everyone, for being in attendance. And if you joined us last night at the Alumni Awards, it is great to see you again. I do have to give one special shout out to my mom, Grace Harrison, joining us live from Tampa, Florida. So that, woo! But to everyone who is joining us live online, thank you so much. We're happy that you are with us, everyone in person and everyone online with us today. So my name is Jessie Pung. I'm the Senior Director of Development in the College of Natural Science. I think this is month 7 for me. This morning, we come to celebrate the spirit of lifelong learning and exploration that defines our college community. Now in our 17th year, Classes Without Quizzes has been a unique opportunity for alumni and friends to reconnect with the intellectual vibrancy of our college and to engage with the latest developments in science and research. Throughout the day, you will learn from leading faculty covering a wide range of fascinating topics spanning the sciences. Whether you're a seasoned science or just curious about the latest advancements in STEM, there is something for everyone at Classes Without Quizzes. So as we embark on the journey of exploration today, let us embrace the spirit of curiosity and discovery that defines our college, the College of Natural Science. Before we officially get started this morning, we have to begin with some housekeeping details. Today's event schedule is in your blue book, so if you're here with us in person, you should have received a blue book. If you didn't, we're totally happy to get you one. And the event schedule is in that blue book, and you can follow along with each presenter. If you have any requests or any questions about the program, you want to ask the woman of the hour, Melody Vinke. So she's in the back left with the laptop, and you can ask her, you can give her your questions, and she can assist you with anything. We want to make sure any curiosities, any questions you have can get answered. I also want to point out our advancement team. Some of you guys know because you've been with us probably for 3 days in a row now, but we've had quite the big weekend at the College of Natural Science. So if you are on the NatSci advancement team, please raise your hand. Let's give a round of applause for this group of people who put this weekend on. Additionally, if you need anything, please go to any of the people that raise their hand and they can be a resource for you. After each of today's presentations, there will be an opportunity to ask questions, like I said, that Melody could help you with, or for those attending virtually, you can submit your questions via Zoom. Our intent is to wrap up everything by 12:30. For those that are with us in person, so everyone I'm looking at, you do have one final exam after today's session, but that exam takes place at the dairy store. So we hope you head over there and you challenge yourself to eating ice cream with the coupon in your blue book. Please turn off your phones or put them on vibrate so we can make sure we have a minimalized distraction event today. At the event's end, there will be a box available on the recycle table, on the registration table to recycle your name tags. And one last thing before we officially get started. Here at the College of Natural Science, we have a wonderful program called the Dean's Research Scholars. These are a group of remarkable undergraduate students conducting high-level research. They are selected for this program and then serve as college ambassadors and are challenged to present their research to the masses. I'd like to ask our 25-26 Dean's Research Scholars to please stand. Over the past 6-7 months, I've had the personal pleasure of interacting with these amazing students, and I can tell you guys, they are our future, and we're so thankful and proud to have them as our scholars for this year. Thank you, guys. You will hear, they deserve it. Let's give another round of applause. You will hear from 2 of these wonderful students giving presentations on the amazing research they are working on later today. During the break, they'll be available, and we encourage you also to talk to them and hear about their work and hear about their journey ahead, as a lot of them are graduating very soon. And now it's my distinct honor to introduce our esteemed Dean of the College of Natural Science at Michigan State University, Dr. Eric L. Hegg. Dean Hegg's journey with MSU began in 2006 when he joined the Department of Biochemistry and Molecular Biology. Prior to his deanship, he served as the Associate Dean for Budget, Planning, Research, and Administration in the College of Natural Science. Dean Hegg's research lies at the intersection of chemistry and biochemistry, focusing on environmentally significant processes such as biofuel production and enzymes involved in the global nitrogen cycle. His commitment to mentorship is evident, having guided nearly 100 students and postdoctoral researchers, including over 50 undergraduate students. Please join me in welcoming Dr. Dean Hegg.
All righty. Well, thank you all for being here, and thank you, Jesse, for the introduction. Good morning, and welcome to Classes About Quizzes, one of our favorite traditions here in the College of Natural Science. And it's really a lot of fun, and I hope you all enjoyed as much as I do. This event is a chance for us to step back into the classroom in a different way, to hear directly from our faculty and students, and to experience the kind of research and discovery happening every day here at Michigan State University. Our goal today is simple. It's to share some of the incredible science taking place and to give you the opportunity to learn something new while enjoying your time with us. This year's program is centered on the powerful idea that is shaping the future research of health sciences here at Michigan State University and in fact around the world. And that's this idea of One Health that you may have heard about. At its core, One Health recognizes that the health of people in the environment, in the plants and animals cannot be disconnected from one another, that we are all connected in this planet, our island home. Through our One Health efforts here at Michigan State University, we're bringing together experts across disciplines, and we're collaborating in new ways, accelerating discovery and preparing the next generation of scientists and health leaders. The challenges we face, whether related to infectious diseases, food insecurity, or environmental change, they do not exist in isolation. As I said, they were all interconnected. And because of that, the solutions that we find to these vexing problems cannot exist in isolation either. They require the collaboration across disciplines in new ways of thinking, bringing people together, people from different disciplines that come together to solve a common problem. This highlights the important role, I would say the essential role, that NatSci plays. Our faculty are leading in the basic research, and they help us understand how these systems function and how they interact with one another, providing the groundwork that is needed for the new innovations that will improve health and quality of life throughout the globe. Today, you will hear from 3 distinguished faculty members whose work brings this idea to life. They were selected for their interdisciplinary approaches and their leadership in understanding the connections between human, plant, animal, and environmental health. I have no doubt that their presentations will be both engaging and inspiring. Through their work, you will see how the basic science that we're doing here in the College of Natural Science drives real-world solutions and impact. You'll hear how researchers are uncovering the connections between living systems and how those systems respond to change, and how that knowledge then leads to better outcomes for the communities here in Michigan and, in fact, around the world. In addition to our faculty speakers, we're also proud to showcase our talented and dedicated deans, research scholars, as Jesse already noted. These outstanding undergraduate students represent the next generation of scientists. Their curiosity, creativity, and a commitment to discovery are already making an impact. And today you'll have the opportunity to learn more about them and their work. We hope you leave today not only with new knowledge, but also with a deeper appreciation of everything that's going on here in the College of Natural Science and how we are shaping the world and making a better tomorrow. Thank you again for being here and for your continued support of the College of Natural Science. And with that, let's get started with the speakers. So to get us started, I'm pleased to introduce Dr. Gina Leinninger. Dr. Leinninger is a Red Cedar Distinguished Professor in the Department of Physiology and in the Neuroscience program. She's also the Graduate Director of the Molecular, Cell, and Integrative Physiology Graduate Program. The Leinninger Lab focuses on its work on neurophysiology mechanisms by which neurons regulate energy balance. Today, Gina will discuss what happens when those mechanisms that regulate so much of the human body are thrown into dysfunction, contributing to the development of obesity and other metabolic diseases. Obesity and chronic pain are prevalent, costly health problems that frequently occur together but have few effective treatments. The Leinninger research team studies how brain cells regulate body weight and pain and how this regulation can be disrupted and how we can modify them to simultaneously support weight loss and pain relief. Understanding this biology can lead to approaches to safely treat and alleviate the suffering of the millions of individuals who suffer from obesity-induced pain. Please join me in welcoming to the stage Dr. Gina Leinninger.
Thank you so much, Dean Hegg, for that very warm introduction. Let's see if a scientist can get this thing going. There we go. Almost there. Sorry, it's being a little fussy for me. Almost there. There we go.
You guys are.
Just takes a minute. It is morning time. Good morning, Spartans. It is wonderful to be with you this morning. I'm so delighted to be here. I am coming to you I work in this building, the ISTB, or Interdisciplinary Science and Technology Building. This is it by day. Here it is by night, where we work assiduously to understand big health problems that impact our world. And as Spartans, we are charged with trying to improve those big health problems. And where better to do that than the College of Natural Science? So the health problem that my team is very interested in addressing is obesity. Obesity was officially declared a disease in 2013 by the American Medical Association, and it is because it has been steadily rising. And this map here shows you the rate of obesity in the United States, across states, over a few decades. And as you see, it's continuing to go up and up, such that in just a few short years, nearly half of the population will be considered to be overweight or obese. Now, this is not an aesthetic issue. What we know is that obesity shortens life and it decreases quality of life. And this is in part because it increases the risk for individuals to develop many secondary complications and diseases, such as things like type 2 diabetes, high blood pressure, cancer, heart disease, and less recognized but truly clinically relevant chronic pain. We know that most individuals with overweight and obesity are suffering with pain that is not well addressed. And so what my team would like to do is to understand how this occurs and to identify other treatments for obesity and obesity-linked pain. Now, there are some great therapies out there. Concern is that they may not be working for everyone. So what do we have for individuals, for half of the population nearly, suffering with obesity and pain? Well, there are things like bariatric surgeries and gastric banding, and you've probably heard of these, and these can have really important weight loss effects. However, not everyone is a candidate for these procedures, and they also have lifelong changes to individuals that may not be tractable for them. Another thing we've learned with these procedures is that often the weight loss does not last long term. The weight comes back. And also that individuals who undergo these procedures are at higher risk for developing alcohol use dependence and suicidality. And in fact, now surgeons will tell individuals who elect for these procedures that they should not plan on ever drinking alcohol again because the risk is so high. So if you're like me, if you like to have an occasional nice glass of red wine in the evening after a hard day, maybe this is not the right procedure for you. What else do we have to help individuals? Well, we have pharmacotherapies, and I bet you've heard some of these. They've gotten very popular, particularly the GLP-1 receptor agonists. So these are things like Ozempic and Wegovy. I'm sure the ad campaigns have told you about these. Now, these can be really powerful. They've only come online in about the last 10, 15 years, but these can be life-changing. They can help individuals who are overweight, who don't have type 2 diabetes, or who do lose pretty substantial body weight. Now, unfortunately, though, these aren't for everybody either. They do produce weight loss as long as you're taking them, but as soon as you discontinue taking these drugs, the weight all does come back. So this would be lifelong therapy. It's expensive therapy. The prices are coming down, but it's just not available for many individuals. And additionally, it has some pretty difficult side effects that often will prevent people from being able to stay on these long term. Nausea, GI distress, and other things. And so these aren't working for everybody either. The other thing we've got is good old lifestyle modification, i.e. diet and exercise. And I think we all know how that feels. Nobody likes to diet. Exercise is good for us, but it's hard to maintain these. And they also often don't work long term in that most dieters do regain weight. So we have therapies, but we need more. We need to recognize that obesity is a heterogeneous disease. Like cancer, it's not all one thing. It's caused by different things, and we need to have a range of treatments that will be available for the range of people experiencing obesity and its comorbidities. Now, that comorbidity is a real issue in itself, in that all of these treatments help individuals to lose weight, but they don't get at the pain. So as I told you, most individuals with overweight and obesity are experiencing chronic pain as well. Now, these individuals looking for pain relief, some of them will take opioids. And in fact, individuals with obesity account for about 14% of the opioid usage in the US. And problematically, opioids can lead to dependence or addiction, and they often don't really help with the significant chronic pain that goes along with obesity. The problem is non-opioid medications, which are out there, actually will promote a bit of weight gain. So if you're an individual and you want to lose weight, but you also manage your pain during that time, taking a medication that actually is going to thwart weight loss is problematic. So we reasoned that an ideal therapy would be one that could help individuals to lose weight and most optimally could alleviate their pain at the same time. And so I like to think of this as no gain, no pain. That is the goal. Now, is there a way to find such a mechanism? To do so, we reasoned in my team that we had to go back to the brain, which regulates body weight and regulates the perception of pain, and understand how it works. What is in there that might be able to do these two things, regulate body weight and pain at the same time? Now, we know that the brain regulates these things, but we also know that the brain is very complex. It has a lot of different kinds of neurons that do different things, and we can't tell them apart from each other easily. So what my team has done is we use genetics to be able to go in and find specific novel types of neurons in the brain that people couldn't see before. And we do this in mice because they can feed and eat and exhibit weight gain or weight loss. So we can track how those neurons actually modulate the body weight and behavior. Now, in particular, I'd like to tell you about how we've used this strategy to identify a new and exciting group of neurons and a signal called neurotensin that I think has the capacity for both no gain and no pain. Now, what is this neurotensin thing? Neurotensin is a small peptide or small protein, and it's expressed by certain neurons within the brain. You can think of neurotensin kind of like a key. Now, any good key can only work if it can fit into its lock, right? And so neurotensin can only work if it can fit into its lock, which is binding to a receptor called neurotensin receptor 1. And if neurotensin binds in that neurotensin receptor 1 lock, so to speak, it can usher in many different effects. Now, these depend on where that neurotensin and its lock are in the brain, but this can be anything from regulating body temperature to assisting with learning and memory, and also some physiology that we were particularly interested in toward our grand challenge of dealing with obesity and pain. It has been shown to regulate pain, feeding, and movement, which could be supportive for the no gain, no pain endeavor. But how it did these things and where specifically in the brain it did them was unclear, because up until a few years ago, there weren't good methods to see neurotensin, to see where that signal was, and to see where the lock was, the receptor that it needs to work. And without that, we couldn't figure out how to use this system to elicit these beneficial effects. So our lab reasoned, if this neurotensin thing could mediate no gain, no pain, then the following things would have to be true. It would have to be there would have to be neurotensin expressing neurons that could decrease feeding, body weight, and pain. There would have to be signaling by that lock, that neurotensin receptor, that could reduce feeding and pain. And if true, the big one, that if we could target those systems with drugs, then we could potentially safely reduce both feeding and pain. So if you'll bear with me today, I'd like to take you through a bit of our work on each of these fronts. We'll start at the very beginning, because Julie Andrews in The Sound of Music told me that's where you always should start. So we'll first look at neurotensin as that key. Are there some neurotensin expressing neurons in the brain that could decrease feeding, weight, and pain? Now, as I told you, a lot of the reason people hadn't been able to study this neurotensin thing much before is because the field didn't have the tools to see where neurotensin was in the brain or where its receptors were. And without those, we were going a bit blind. And so to get around that, my lab has made mouse models in which we can genetically label and see all of the neurotensin neurons across the brain, finally being able to see where they are so we could figure out what they might do. And so we can look in these brains and see these green labeled neurotensin neurons, and we noticed that there were a whole bunch of them in this area called the lateral hypothalamic area that is circled here. Now, this area really caught our eye because it had a bunch of these neurotensin neurons, and because it was well established that this brain area was critical for regulating feeding and body weight. So we figured we might be on to something by discovering this huge population of neurons in this area, but no one had ever studied them before because they couldn't see them or find them. So this was a whole new frontier. We wanted to know what they do, and particularly if they could regulate feeding, body weight, and pain. To do that, we used our mouse models that allow us to see those neurons, but also to do exciting things to test if they're important and how they work. So what can we do? Well, first we reasoned these neurotensin expressing neurons are there in this important brain area, but are they really important? And a way to figure that out is if we take them away. If they're really important, if we killed those neurons, we took them away, that should have a negative effect on the body weight. And so we developed a strategy to do that, where we leave all the neurotensin neurons in the brain intact, but only in this lateral hypothalamic area do we deplete those green neurotensin neurons. And this is an example of what it looks like. We retain all other kinds of neurons, but we've depleted just those green neurotensin ones. Now, if we've gotten rid of those neurotensin neurons, does it matter to the physiology of the mouse? Well, it became very apparent quickly that it did. So mice that don't have neurotensin in their LHA neurotensin neurons developed pretty extensive obesity. So that told us that the neurons must be important. We need them to regulate body weight. But how do they do that? The flip side of this argument is that if we need them to regulate feeding and body weight, if we could activate them, if we could make them work, promote them to work, maybe that would help to support weight loss. So we tried that too. We reasoned that if we can activate these neurons, it would reveal what the physiology and behaviors they regulate. So we used a strategy, I will not belabor it, but basically we can go in and we can selectively activate just those LHA neurotensin neurons in the brain. And the little lightning bolt here will indicate the activation. We activate them with a drug, but we're just turning them on. That's what you really need to know. So what happens when we turn those neurons on? Well, mice eat less. They move around more, so both the diet and exercise component, and they lose body weight, suggesting this group of neurons could be a no-gain type of group of neurons. But what about the pain? Again, we're looking for the big dream, the dream of alleviating both the weight and the pain. And so here we use that strategy of activating those LHA neurotensin neurons again in mice, and the activated ones will be in red. Now, if mice don't have any pain, activating those neurons doesn't do anything to the mice's behavior. But this graph is going to essentially map how much pain the mouse is feeling. If we injure the nerve, somewhat similar to what individuals with diabetic neuropathy might feel, and that's relevant because many individuals with obesity do develop type 2 diabetes and neuropathy. If we injure nerves in that way, these mice all experience pain. Now, if, however, we activate those LHA neurotensin neurons, it reverses that pain completely. Now, this was just a few days after the onset of that injury, and we reasoned that more akin to obesity and neuropathy that occurs with it, that's more of a chronic sustained pain. Could we with these neurons, by activating, relieve that chronic sustained pain? And so we let these mice go a little longer. They resume their pain if we're not activating their LHA neurotensin neurons. But if we activate them again about a month later, we can still reverse that pain completely, suggesting these neurons really do have the potential to alleviate severe pain, chronic pain, the kind that might be present in obesity. But the elephant in the room was, you didn't actually study obesity-induced pain. Will it really work in that context? And so we generated obesity-induced pain. We can take mice and either feed them a normal, healthy diet or a high-fat diet that will induce their obesity. We can see that feeding them that diet increases their body weight. They become diet-induced obese. And we also see that these animals experience more pain. When this bar goes down, the lower it is, the more pain they feel. Obese animals have pain, similar to what we know happens for humans. So if we can turn on those special LHA neurotensin neurons, can we alleviate that pain? So we generated these obese animals in which we could selectively turn on just those important LHA neurotensin neurons and looked at what happened. Now, before we turn them on, all of the animals have pain. But when we turn on their LHA neurotensin neurons, it alleviates that pain. As long as we have the neurons on, as soon as the neurons aren't on anymore, the pain is back. Suggesting that these could really be, these LHA neurotensin neurons, an endogenous, a source of brain neurotensin, could be no gain, no pain neurons. Now, if that was true, then signaling via their receptors, by the lock that the neurotensin fits into, that would also have to be able to reduce feeding and pain. And that was really important for us to determine because these kinds of receptors are the kinds of things you can develop drugs to. So we needed to see if the receptor, the lock, could do the same kinds of things. Now, here's where the brain is enormously fun to study, but very complex. It is an enormous puzzle. So these neurotensin neurons I told you about, they make this neurotensin key and they release it to many different areas of the brain that all do important things. But we wanted to find the areas of the brain that have the receptor, the lock, and that might modulate both feeding and pain. And so we zeroed in on two areas, one, the ventral tegmental area here in orange, and another in blue, the lateral preoptic area, because these areas have that neurotensin receptor. They could respond to neurotensin if they get it, but what do they do if they get it? And so we really wanted to know, does neurotensin signal via those areas to regulate feeding, body weight, and pain? Now, to test this is a little trickier. We used our mouse models, but here we wanted to only activate the neurotensin neurons that projected to the VTA, not all the other projections that they go to. So we use this very cool strategy where we make it so these neurons, if we shine blue light on their terminals, it will activate just that pathway. So we can see only what that one does. And so when we activated that pathway just to this ventral tegmental area, what it did was nothing to feeding. A bit of a bummer, but not every scientific discovery is always the one you think it's going to be. It didn't regulate feeding. But what it does do is it does reduce pain. So in normal weight mice, they don't have any pain. Obese mice do have pain, but turning on just that pathway could reduce that pain a bit. That bar went up instead of down. maybe this is one site where the neurotensin can provide analgesia. But it didn't reduce the feeding, so we had some more work to do. So we looked at that other brain area, that projection that went to this area called the lateral preoptic area, or LPO. And we used the same strategy of the blue light to activate just those neurons in their projections. And when we did that, it robustly decreases feeding. In fact, if we turn these neurons on in obese mice, the animals will lose considerable amounts of weight and it will stay off. However, it doesn't do anything to pain. If we turn on these neurons in mice that have pain, it doesn't make that pain any better. What this told us is that neurotensin can modulate pain and gain, but it does it by different brain areas, and that's important. It means we have to be able to have something that can get to both of those brain areas. But that neurotensin key, if it gets to the right lock, can suppress both feeding and pain. So the real question, could we do this pharmacologically? Could we ultimately develop a drug that would help to do this in real life? Now, this is work that we've been doing in collaboration with Lauren Slosky at the University of Minnesota. She developed this new neurotensin receptor molecule called SBI 553, and she's been studying it in the context of drug addiction. And it's shown that this thing can decrease individuals from taking psychostimulants or alcohol. But we wondered, could it reduce natural rewards like feeding? And so to study this, we took obese mice and we made them hungry. And then we treated them with either control or that SBI 553 to promote that neurotensin receptor signaling, and asked, can it help in these obese animals? And what we found so far is that it can indeed reduce their feeding, and it can indeed reduce body weight gain. Now, we have not yet gotten to be able to see if it will alleviate their pain, but we are working on that in the lab right now. So stick with me. I hope to have more exciting updates for you soon. So what it suggests is that these drugs do have potential to safely reduce both feeding and weight gain. SBI 553 has been tested in other types of pain models, and it has been shown to reduce pain. So we are hopeful we are on the right track. So by further studying this neurotensin system, we hope to understand the biology that can safely treat obesity pain, and so that MSU can lead the way in addressing this grand health challenge. Now, none of this gets done without the amazing team, and I've been so happy to get to work primarily with undergraduates in the lab and graduate students, many who I've had the pleasure of graduating over the years. I want to appreciate all the work that they do. My MSU collaborator, Geoffroy Lamé, who taught me all about assessing pain, and my collaborator, Lauren Slosky at the University of Minnesota, and Steve Olson, who provides the SBI, and the federal support that we receive for the NIH and the DoD for this work. And I want to thank you for being here today, for being interested in science, for listening to me prattle on about it. And I'd be happy to answer any questions you might have. Thank you.
All righty. Thank you very much, Gina, for a fantastic talk. So we have time for about 5 questions here, and I'm going to ask that you wait until the mic gets to you so that people online can hear us. We have someone who'll run the mic to you. So
I'm running the mic. Question. Make sure you hold it up.
Amazing work. This is a pretty fundamental question. How does one label neurons?
Great question. So what we can do is we label neurons by genetically tagging them. We make them express this enzyme called Cre recombinase, but you can think of it like a label. And once we have that Cre in there, we can get that Cre to do things like only express a green fluorescent protein. That's how I was able to show you those green neurons. It makes them look green. We can also do things like take those neurons away, activate them, inhibit them. So it requires being able to genetically modify those neurons and how they're made. That's why thus far we're doing those things in mice. We cannot genetically modify people, but we can use mice and genetics to find the right neurons, to understand how they work, and by understanding that biology, then leverage it with pharmacology to be able to mediate those same effects. Great question. It's fun stuff.
Yeah, thank you for the presentation. I was wondering, how does one measure pain? I was interested in that. You made that comment that you learned So I would like to learn too.
Yeah, yes. So I'll be candid that I knew for a long time that neurotensin could alleviate pain, but I didn't want to study it because I didn't want to make the mice experience pain. Letting mice eat lots of food is great, right, for them, but the pain was tough. So how we do that, there are a couple different kinds of pain that people and mice can experience. So there's thermal pain, so we can put them on a hot surface and see how quickly they'll lift up their paws. We can do the same thing with cold and see how much they lift up. We always do it's a short test, so they are not getting injured. But just like you, if you touch something hot or something really cold, you're going to lift your hand up. The mice do the same thing. Where we find neurotensin really helps is not actually in that thermal pain area, but in what we call mechanical pain. So if you touch your finger on a needle or something sharp and you would withdraw your hand, mice will do the same kind of thing. Again, we're actually not trying to injure them. So what we do is we poke their little mouse feet with these little filaments. So they're not sharp per se, but they're you'll feel them. They're of different thicknesses. And so we poke the feet and we see when they'll lift their paw away. And animals that don't have much pain, you can poke them with progressively thicker filaments and they are okay with it. Animals that are experiencing pain will only be able to tolerate being poked with really thin filaments that to you normal mice wouldn't be a problem at all. So that's how we do it.
Thank you so much for your talk. I really enjoyed your work. In the first experiment you did, I saw that you knocked out neurotensin and then the mice ended up becoming obese. I was wondering if you had a sort of control group, which was like the rescue, just so that we can be sure whether the experimental manipulation itself didn't induce the obesity.
You are a scientist. I love it. Yes, controls are the fundamental, most important thing in science. So what we really did there is we actually took away the whole neuron. And so we killed those neurons that express neurotensin, as well as a bunch of other things. So we can't fully guarantee it was the neurotensin in them that was the important thing. To your question, the best experiment would then be to reintroduce that neurotensin and see if that fixed it. We couldn't do it in that experiment because we'd killed the neurons. We couldn't put the neurotensin back because there was no neuron there to hold it. What we're doing right now is we've created mice in which we can delete neurotensin just from those neurons, leave everything else about the neuron intact, all its other signals, and see if that does something to the body weight and restore the neurotensin to those mice. That data is still ongoing, but what I can share with you guys is so far it looks like taking the neurotensin away does all the things we might have expected it to. The mice gain weight and they have increased pain. We are not to the point where we've been able to restore that neurotensin yet, but we anticipate that it will alleviate both of those things. Great question. Come visit the lab.
Hi, that was a great talk. At the beginning of your presentation, you talked about other effects besides pain and obesity, and I'm wondering if there are studies that are ongoing about the effects that might be helpful in other aspects of healthcare.
Yes, absolutely. So we know that some of the current treatments for obesity, the bariatric surgery and the GLP-1 agonists, they are very helpful for reducing cardiovascular concerns. They've been particularly the GLP-1 receptor agonists. Well, the other effects, I think we're still too early to see the things like cancer. We don't know yet, but we do know that once the weight is lost, it helps to improve many of these things. Jury's out on whether the pain goes away with those treatments, and that's why we're looking for something else. And so, we're really early actually in those treatments about how they all work for these secondary complications. So we're still finding that data out.
That was really nice, Gina. Thank you. I'm going to ask that question sort of on the opposite side, which is one of the raps I've sort of read about on the GLP-1s, and then I could see it potentially in this too, is that you're altering sort of activities that are driving people's behavior. So it's great if they're eating less, but they may be doing other things less as well. And so is there anything that you're looking at in the neurotensin? Because probably there are other aspects of neurotensin besides just pain and
That's a
feeding as
great
well.
question. People are going to think I paid you to ask that
No,
question.
no, not at all.
So I didn't get to share it with you today because they said I couldn't talk for 3 hours. But we've also been really looking to see if this neurotensin thing could do things like promote too much reward or too little reward. Again, knowing that some of these manipulations can promote substance abuse, we wanted to make sure we weren't developing another strategy to do that. So what I can tell you is that modulating these neurotensin neurons in the system actually, it doesn't seem to be rewarding to the effect that it would promote substance abuse or intake. But neither is it aversive, meaning that the animals aren't eating less because they feel crummy and don't want to do anything. In fact, they seem to move around more and feel great. They're just, the reason they're not eating is neutral. It restrains the feeding and alleviates the pain, but it's neither something that they're going to crave, it's not going to be a drug of abuse, nor something that they would avoid. And that's where I think we're in the sweet spot of this having real therapeutic potential because it's a neutral drug with beneficial effects. Did that get at your question?
We have an online question. So I'll ask that on their behalf. Have you assessed how long the pharmacological treatment could keep pain reduced?
That is a great question. We are on that track. So first we had to do short-term studies to see, if it could work. Now we're entering into those longer-term studies. From prior work from a collaborator who studied, again, non-obesity types of pain, they've been able to show that keeping this drug on board for several days can alleviate that pain consistently for several days. Whether that will be true in obesity, we have yet to see, but we are doing those experiments right now.
All right, with that, let us thank Gina once more time. All righty. Thank you, Gina. That was truly fascinating. So, as you all know, a key component to the college's undergraduate educational experience is, in fact, research. And we are, in fact, committed to advancing knowledge by providing progressive undergraduate research opportunities. Whether it's developing research plans, writing proposals, conducting research itself, analyzing data, or presenting research results, these opportunities provide students with a deep understanding in a scientific area while also gaining hands-on experience, basically training the students to understand what it's like to be a scientist. Undergraduate research is a great way for students to learn from faculty, researchers, graduate students, and other undergraduate students who share similar career interests and goals. Simply put, being involved in research greatly enhances a student's degree while providing them with the experience needed to make them competitive for graduate school or other careers. Recognizing the value of having these students communicate their research and campus experiences and to be able to describe to other students as well as alumni what it's like to be a student here at MSU today, the college established the Dean's Research Scholars program in 2012. The Dean's Research Scholars personify the high caliber of the college's student body and exemplify the traits of true Spartans. That is, hard work, intelligence, perseverance, and problem solving. And we were honored to have these students represent the college this past year at events and speaking engagements like this one. So to help give you a glimpse of what it's like, one of our Dean's Research Scholars is here today with us, who will be talking, and his name is Arkesh Das. He is a biochemistry and biotechnology senior, a Dean's Research Scholar. Please join me in welcoming Arkesh.
Thank you for the kind introduction, Dr. Hegg. Let me navigate to my presentation. All right. But yeah, as Dr. Hegg mentioned, my name is Arkesh, and I'm an undergrad here at Michigan State. And last year, I was accepted into medical school through the University of Toledo's MedStart program, and I'll be starting med school there this summer. So this last year has basically been like a gap year for me. I've been able to explore my interests outside of medicine. I've been able to hang out with my friends and family and finally go to some football games, and really just grow as a person. And traditionally, that's what the purpose of a gap year is. However, as I've been getting ready to graduate, I've been talking with a lot of my friends who would traditionally be applying to medical school right now, and the question they're asking themselves isn't whether or not they should take a gap year, it's how many they need to take to remain competitive. Now, this isn't something that's happening just at Michigan State. Nationally, these last few cycles, nearly 3/4 of all incoming med students will have taken at least one gap year. In 2017, that number was about 60%. So on an individual level, taking a gap year seems like a personal choice. However, the choice to delay one's medical training doesn't just affect the individual, because the public directly invests in the training of future doctors at multiple different levels. This year so far, the federal government has spent about 30% of its budget on healthcare. So roughly about 1 in 3 taxpayer dollars has gone towards funding medicine and medical education. And I'm sure I don't need to tell you guys this, but medical school is expensive. But despite this, only about 5% of medical school revenue comes from tuition and fees. About 20% comes directly from federal funding. Now, once a student finishes medical school, they aren't a fully fledged doctor yet. They still have to complete their residency training. And each of these almost 120,000 residency spots are directly funded by the federal government to the tune of $21 billion each year. So medical education isn't just an individual investment. It's a public investment at every single stage. And the return on that investment is the number of years that a physician ultimately serves the public. And what we see is that as the completion of training is delayed, the number of physician years you get out of each doctor declines. Between the most recent graduating classes of medical students and those who are currently in medical school, the number of physician years out for each doctor has dropped by about half a year. Now, that doesn't seem like a lot. It's about a decline of 5 months. But when you scale that across the over 100,000 students that are in medical school right now, you lose roughly the equivalent to 40,000 physician years of service. And for every year that the average graduation age will continue to increase, you're going to lose the equivalent to 2,900 full primary care careers from the workforce. So far I focused on the direct loss of physician years from delayed entry into practice, but there's also a hidden indirect loss in the form of burnout. Now, I'm sure you guys have heard about the burnout problem, but in primary care, nearly 60% of physicians currently report at least one symptom of burnout. And importantly, these are physicians who trained before gap years became this widespread expectation. Now that we're seeing students spend more years in a sustained high-pressure environment competing with each other before they even start their training, what we're doing is we're setting these people up to burn out before they even start to practice. And when a physician burns out, the public doesn't just lose out on a couple of months. We lose an entire physician's career and the decades they would have served the public and their communities. Speaking of communities, medical schools have long said in their admission statements that they want to emphasize training physicians who will serve underserved and rural communities. And across the 176 MD granting schools in the US, over half of them, including Michigan State, explicitly reference diversity, and many specifically mention service to underserved or rural populations. But here's the thing, who can actually afford to take gap years? It's not students that need to start earning right away. It's not students who have families to support. And it's not the people who don't have the option of living at home rent-free. We say we value service to underserved communities, but what we've done is essentially created a socioeconomic filter that favors those who can afford to delay and filters out those who can't. Similarly, since about the 1980s, we've identified that there will be a shortage of primary care physicians in the US. So for decades, many medical schools have said they want to emphasize training primary care physicians. But if that's the case, then why does primary care still remain one of the least competitive residency pathways? Last month during the match, there were over 1,600 primary care residency spots that went unfilled. So there's clearly a disconnect here, right? The factors that we currently use to admit medical students are not actually selecting for the kinds of applicants that these mission statements describe. Now, this whole time, I've been saying that students feel that they need to take a gap year to be competitive, and I want to pivot this last half of the talk by offering an explanation as to why this might be happening. In the most recent admissions cycle, there were about 54,000 applicants, and only 43% of them had the chance to enter medical school. In economics and game theory, this is something that you would call a zero-sum game. Because there are more applicants than there are seats, for every one person to be admitted into medical school, someone else necessarily is not. And in a zero-sum system, applicants are incentivized to compete with each other in any way possible in order to get an edge over their peers in their competition. This has led to what I like to call the pre-med arms race. I'm sure you guys have all experienced this from your friends or your family that are in the medical field right now. Students are accumulating more and more clinical hours, more research, more volunteering, and just more of everything. And within that system, taking a gap year becomes a temporary competitive advantage. But here's the thing. As at first, maybe a few applicants take a gap year to build hours. But then once others see that, they start doing the same. And as more and more people take a gap year, that advantage disappears. And so the baseline essentially shifts. Now what people are doing is, it's not about whether you take a gap year or not, it's how many you are willing to take and how many things you're willing to pile up on your application while you're doing them to build up your resume. But that raises the question, is this escalation actually going to make better doctors? Or are we just sacrificing years of service for a system that's becoming increasingly more inefficient? Now, this is a problem that I've thought about pretty much ever since I started my pre-med journey, and I don't think I'm alone. From talking to my friends, my professors, and physicians who were, or do teach pre-meds, I think this is something that has crossed everyone's mind at some point. I just happen to be a data scientist, which means I'm a weirdo who likes to look at numbers. And because I was accepted early through Toledo's program, I was able to use this last year to reflect, write down my thoughts, and publish my first paper in MSU Student Journal. But the reason I'm speaking with you all today is because I think this is something that people just don't really talk about. It's almost like an open secret in pre-med culture. Everyone feels it, everyone experiences it, but very few people actually stop and question this. And while I was in the thick of it, going through the pre-med arms race and everything, I didn't question it either. So my goal here isn't to say that I have all the answers. I am, after all, just one person, but I really want to start a conversation that I hope will reach the people who have the power to change this, namely admissions committees and medical schools themselves. And right now, I think this is an especially important moment to be having this discussion, as Michigan State is actively restructuring how health education is organized through the One Health initiative. When a system is being redesigned like this, I think that's when questions like this matter the most. So, because if we select and train physicians in a way that unintentionally filters out groups or shortens their careers, it doesn't just affect the individual applicants. It shapes the long-term health of the communities and the communities that these physicians will serve. And at the end of the day, this isn't just an applicant-driven problem. It's a system-level incentive problem. This increase in gap years is just a symptom of the current metrics that we use to evaluate who does and doesn't get into medical school. So the question becomes, if the incentives are driving outcomes we don't actually want, how do we redesign the system to produce the doctors we want without wasting years and talent in the process? Thank you.
All righty. Thank you, Arkesh, for that thoughtful and thought-provoking talk. Any questions here from the audience or online?
Great talk, Arkesh. So you talked about solutions. I was wondering if you had any potential solutions in mind, because this seems like a very passionate topic to you.
Yeah, you're absolutely right. I mean, I've been thinking about this a lot. So, like I said, I mean, I am just one person, but some potential solutions that I would be, and in case I haven't made it clear, I don't think that gap years are inherently a bad thing. I mean, like I mentioned, this last year has sort of been like a gap year for me. I've had some time to relax and pursue things outside of medicine, and I know that a ton of people come into medicine from other careers, and I think they bring a very valuable and unique perspective. So I think a solution isn't something that would eliminate gap years, but make it so that they aren't the default way that people are competing with each other. So realistically, you can't control the demand of medicine or the number of people who want to become doctors, but you can change how they are competing with each other. So right now, I think we're essentially treating time as a proxy for merit. And so one way to address that is to normalize the advantage people get from gap years. So that could be things like capping how many extra research hours you can accumulate or clinical hours, and being transparent about that so that people are taking gap years for the right reasons, to grow and to develop and to get a unique perspective. Another thing too, I mean, I might be a little bit biased here given that I got in through an early assurance pathway, but I think expanding these pathways could help, because when a student already knows they have a spot, they're less incentivized to win or try to compete with each other through this admissions game. And I think programs like Toledo's or Michigan State's OMSP program can be used in that way to control that competition. Northeastern Ohio also has a medical school, and what they do is they allow students to matriculate either immediately or defer their acceptance. So if they need to start their career right away, they can, or they can use a year to grow and to develop. And finally, I think another solution I've thought about, might be a little bit controversial, is maybe shifting back towards more standardized metrics like GPA and MCAT, because while they're not perfect, they're at least things that everyone can compete on regardless of their background. And it's not just about who has more connections or who can get all these opportunities. It's a standardized thing that everyone can compete on and be on an equal footing with. But it's not perfect, but I think it might be fairer than what we have now.
Thank you. Other questions? We got one right over here.
You're giving me flashbacks back to 1978 with the war between students. I still remember that. But you know, one point I guess I'd like to make is, once you get into medical school, that's fine, but most of the burnout is coming from pre-authorizations, staffing problems, overwork. How can you have a family when you've got $100,000 in student debt? And you constant worry, constant pressure to keep working. You know, there's more to this. And of course, back in 1978, I'll just tell you, nobody had a concept of gap years. There was pressure to get through your 8 years. You know, so.
Yeah, I mean, I think that's a very important point, and kind of what I mentioned in my talk is that, like, right now, even the doctors that trained, 20, 30 years ago, they're experiencing high levels of burnout. This is essentially another layer on top of that. And so I would predict that, yes, there's problems right now that are causing physicians to burn out, but if you have doctors that are entering that system already burnt out, already going through this competition, I think that's just going to make this problem even worse, and why it's something we need to address.
Your presentation was really good. I'm a little confused. So is your gap year to build your resume, or is your gap year to actually make you a better doctor down the road?
That's honestly the question that I asked, right? Is that these days, especially from speaking to like my friends and stuff, people are taking gap years because they feel they need these hours to compete. And I think that's completely different to an ideal gap year where you're spending time exploring and gaining a unique perspective. So I think that if you make it so that gap years aren't an aspect of competition, but something people choose to take, then you can get back to where people are taking these gap years or doing other things outside of medicine to gain a valuable perspective. I think that the current state of gap years and the reason people are taking them doesn't incentivize that.
So if I'm looking for a new doctor, should I ask them how many gap years they've taken?
You could. I mean, it's a pretty new trend. So maybe 10, 20 years, when everyone who's in this current admission system becomes a doctor, you might be able to figure that out.
I know we had a question over here.
Yes, my question is not necessarily in gap years, but this problem with physician burnout. Does this exist strictly in the United States, or does it exist in other, like Europe, Canada, other countries?
Yeah, I'm not exactly sure about that, but I imagine it would, especially with the COVID-19 pandemic and everything like that. I think that definitely, from my research, I saw that really did increase the level of burnout and the things that people were feeling as physicians. But I would have to look into that. I'm honestly not sure how this affects other countries as well. But one thing I would mention is that a lot of countries have a different medical training system. So exactly. So those students, they find out in high school whether or not they can be a physician, and they train for 6 years rather than having an undergraduate degree, then a medical degree. I think that's a conversation that's worth having. I think there's pros and cons to both sides.
All righty. Do we have time for one more question? We have time for one more question.
We have an online question here. So the question is, how do you think that this research will help you in the future to avoid burnout?
Yeah, I mean, I guess, I don't know if it's necessarily the research, but this year that I've spent, I think I've been able to mature and grow a lot as a person. This is research that I really want to continue to pursue as I'm in medical school, and just to keep this conversation going. I think what I have right now is like a solid proof of concept, but this might just be the data scientist in me, but I just really want more data to analyze this trend historically and things like that. So these last few months, I've been emailing like the AMC back and forth to get access to older reports. But ideally, I would love to gain access to like more granular data so I can do stronger analyses and figure out why this is happening for certain. And so, I mean, I'll be heading into med school in a few months, and I love opportunities to collaborate or connect with someone who has that level of access, because the more detailed the data, the better I can analyze long-term trends. But I think this has been a very impactful project to me. The research I did previously was nothing related to this stuff at all, but I gained a lot of valuable experience through the research I did, like in the biochemistry department and in the department of psychology. But yeah.
Arkesh, quick question for you. Did the paper that you talked about you published, was it on this, or was it on something completely different?
It was on this. That was my first time publishing a paper all by myself, and I wouldn't have been able to do it without the help of MSU's like student journal, and so I think that was really valuable. But yeah, it was on this research.
So if people want to learn more about it, they can go and find your paper, and
yeah.
Just all written down. Okay, excellent. Well, join me in thanking Arkesh. All righty, so next we're going to hear from Guanyu Hu, an associate professor in the Department of Statistics and Probability. His research focuses on the development and application of statistical methods to complex real-world data, including sports analytics, environmental statistics, educational psychological measurements, and health sciences. Dr. Hu also works with spatial omics data, building hierarchical and spatial structured models to better understand biological processes at high resolution. It's like building a map of biological processes to better understand them, expanding foundational knowledge of our world to answer our most pressing questions. Please join me in welcoming Guanyu Hu.
Okay, thanks our Dean's nice introduction. I'm very happy to give a talk in this kind of very great event. So as you know, I'm very new here. So I just joined the MSU last year. So that I think this is my first biggest event at MSU's. So today, I'm from the statistics background. That is a little bit from the others, from the biology, psychology, other fields. So, but the one thing I noticed from our previous talks, they all talk about a lot of data. So first, I want to do one advertisement for myself and for our department. So that is, if you think your data is complicated, you cannot do a valid model for that, come to see us. And also, if you think your t-test is not that powerful, come to see us. If you want to explore some causality instead of correlation, come to see us. Okay, so today my talk is about this kind of genomic studies. So I'm not very good at the genomics, but I would like to explain how the statistical model help us to explore some genomics pattern in this kind of real-world data sets. And also, first, my title may be a little bit complicated. That is a lot about Bayesian, Nonparametric, Crosson. Maybe that is some unusual words. So hopefully my slides, I try my best to reduce the formulas in my slides to help you to understand what my talk is going on. Okay, this is the content of today my talk. First, I will briefly introduce this kind of very hot and popular data set recently that is called spatialomics data. And then I would like to discuss what is my statistical model, and then discuss how can we do the computation. Maybe that I will ignore that since that part is too statistical, and then I will discuss my simulation study and the real data application. Okay. Let's move on our first session. That is our transcriptomics data evolution. Since in this screen I cannot see my slide, so I just can explain this. So I think that is spatialomics data is very new technology that is emerging maybe last 3 or 4 years. So, but previously I think a lot of you know what is this kind of RNA sequence data or this kind of omics data or genomics data. So this is a major differences between this kind of data set. And the first of the data that is we call the box RNA sequence data. So that is like the smoothing. So this kind of data will have all the fruits together, so that will mess up all this kind of sample. So we know exactly what genes are expressed, but we lost all this kind of spatial context. So that is this kind of bug data. I think that is this data is analysis maybe back to last maybe 20 or 30 years, and then people move on. Not only for this kind of bug data, they can detect some data set from the cells level. So this kind of genomic data becomes to a single cell RNA sequence data. So I will treat this data as the fruit bot. So that is we isolate the individual cells. And then we can, for each cells, we have this kind of RNA expression. So that is we know what each cell is doing, but still do not know who is the neighbor who we are. So that is this kind of data. And recently, I think that this is the spatialomics data is the cover this kind of method in 2024 that the Nature Methods that journals. So this kind of spatial resolute transcriptomics data, we can think about that is the fruit dot. So we sequence this kind of RNA while preserving this kind of exact 2D or 3D coordinates of this kind of tissue. So that is we can RNA, not only we can observe this kind of RNA expression, we also can know this kind of RNA expression is where this kind of RNA expression is spatially distributed. So that is data looks like. So this is the plot how people get this data set. For this kind of figure A, we will find this is our tissue. So and then we can have, based on the tissue, we can find this kind of image-oriented spatial coordinates. So that will be this kind of plot B. And then we can extract some this kind of spatial spot from these kind of tissues based on different resolution. That is same like if we have certain picture, each picture will have some pixels. So each pixels we can observe some color, this kind of scale. That is same like what we have for this kind of spatialomics data. Then for each pixel we can have some our techniques to get this kind of RNA expression. So that is this kind of screening this kind of technology. We can get this kind of RNA expression. And then we can combine this kind of two part of data together that will give this kind of spatial omic data. So that is kind of spatial omic that comes from. So we have RNA expression, and then we have its own spatial coordinates. Okay, so what we need to do for these studies, since that is in this kind of RNA expression data, we need to do some analysis. So what we would like to do, that is this kind of biological tissue are not randomly soups up cells. Since that is, we cannot just treat them at randomly. Since the reason why they have this kind of spatial distribution, they have some meaning. So they are highly structured. So what we want to do, that is, we want to do some tissue architecture. For example, a brain has distinguishing and matrix layers. So a tumor has its core. So we want to find which region is nearby have a similar pattern. So that is, we want to find a certain boundary, and also we want to have some surrounding immune cells. So that is what we want to do for this kind of spatial cluster. And so based on this kind of spatial cluster problem, we want to identify these kind of spatial regions. So that means we want to see which kind of, where these regions are most similar. We can treat them together and do some similar treatment or analysis. So that we want to automatically group the spots into clusters that represent their underlying biological structure. So that is what we want to do. So that is, if we just treat all these kind of spatial location randomly, so that is, we need to maybe consider maybe n different subgroups. But if we can group them together to maybe, for example, to the k groups, what we can do, that is, we can just do maybe the k data analysis. And another thing that is, this kind of genes also are spatially varying. So we want to find some specific gene that drives the formation of these spatial domains. Since for some this kind of disease, not all the genes are works. So we need to want to identify which gene is our differentiated genes. So currently there are a lot of methods doing this kind of stuff. For example, I list our methods. So, and also this kind of method is popular in a lot of top journals like Nature Communication, Nature Biotechnology, and Nature Methods. But this method, there are a key problem for this kind of method, that is, this kind of RNA sequence data is very high dimensional. We will see for the gene expression, we have a lot of these kind of different genes. So this kind of high dimensional sequence data is messy. To handle it all these methods need to reduce dimension reduction method like the PCA or negative matrix factorization to compare the data before the clustering. So in their method, there are two steps. First, they do the dimension reduction. Then, based on this kind of reduced model, they do this kind of clustering analysis. But compressing this kind of data, they lose some original count structure of obscure down real biological signals. So that is some issue from this data set. And also, previously, they did some research and they do some transformation of the original data. So as you know, that is RNA sequencing data. Originally, that is by this kind of count rate. So they will do some transformation, normalize them, so that will use some normal distribution to do that, but they will lose some information. So our proposal, our method called the BMP space. So this BMP space is a clustering framework for SRT data based purely on original count rate. So first, we do not do any transformation of our data. We just use our original read count. And the key contribution of our models, first step, that is, we directly model the counts. So that is, we do not do any pre-processing of our data set. We do not do no normalization, standardization of the original, this kind of RNA sequence count. The second thing, that is, since we're working on this kind of high-dimensional data, so not all the genes are useful for our original detection. So now what we want to do, that is, we want to do internal variable selection. So that is, we can automatically identify which genes are actually discriminated spots or separating them from the background noise. So that is, we do certain this kind of differential analysis of these different genes. The third step, and also a very important step, that is, we do the alternatively and spatially availed. So that is, we employ a specific statistical framework we call the Markov random field mixture of finite mixture model to determine the number of clusters automatically and also enforcing some spatial smoothness. That is our goal. So if you're familiar with some clustering analysis, a lot of clustering analysis, you need to specify number of clusters in advance, but our method, you do not need to specify number of clusters in advance. Okay, let's move on our model setup. I try my best to reduce my formulas. So the first step, that is, for the RNA sequence data, there are very common issue, that is, for this kind of RNA sequence data, since if we deal with this kind of raw count data, you will observe a lot of zeros. So this kind of SRT data essentially have a mass separation of spots and genes, where there looks at this metric, we can see a lot of zeros in that data set. So that is, for this kind of zeros, we should deal with these zeros. So then if we do some traditional count data models, they will not very good at this kind of many zero count. So there are two distinct suspects. The first zero, that means this is a biological zero, that is true zeros. So that is the gene are generally turned off in the specific cell types. So that is true zero. And also there are some technical drop-offs. So the genes are automatically expressed, but our sequencing technology physically miss it during this kind of capture. So that is based on these technologies. So now, how can we deal with data zero? So idea that is, from the count data, if you go to Stat 101 course, the professor will tell you can use the Poisson model to deal with this kind of counter data. So the Poisson model is natural distribution for this kind of counter value data. So, but why not just use the standard, this kind of bell curve or normal distribution? So the counter data usually model with a Poisson distribution, but a standard Poisson distribution cannot handle mass this kind of zero counts. So to fix this, our first step is to account this kind of zero inflated Poisson model. So that is, we add a certain probability on this kind of zero observations. So that is our techniques. So our techniques, that is, we construct this kind of zero inflated model. So this zero inflated model, we have two parts. The first part just comes from the original Poisson that will follow this kind of Fyi. This is still our Poisson model. But another part, that is, we add another term to add in this kind of probability to this kind of zero observation. So that is our idea. So this is our, we just have the two-way mixture. One, we inflated this kind of Poisson. The other one, we just contribute from our original Poisson model. This is our first step to discontract this zero inflated. And the second thing, that is, we still want to model this kind of biological heterogeneities. So that is our major goals. So that is, what is this kind of biological function of Fyi? This is a Poisson model, but different cell types in our tissue should have different this kind of expression. That is our assumption. So this kind of key ingredients for this kind of model, that is, we add a certain mixture type of this kind of Poisson model into our studies. So that is, we can, for each different types of the cells, they will have different this kind of count expression patterns. And here, we still do one size factor here. So there are some spots maybe have sequential deeper or better than others. So we have this kind of size factor to adjust the overall loudness of this kind of stuff. So for example, in some social science, if we do some disease mapping model, we always will have this kind of set effect. For example, we will adjust state level this kind of population. That is similar story. And finally, we have this kind of expression level. So the baseline expression rate of gene J, especially inside this kind of class K. So that is our idea. We adjust a lot of things we think that should not be considered in our this kind of heterogeneity of this kind of cell types. And the second thing, that is, still we want to do some feature selection. So the feature selection, that is the simple idea, that is, if this kind of feature should have effects on our spatial cluster, we should include them. If we do not think that is important, we should exclude them. So that is a major idea. So that is, this is a toss coin. If the coin is head, we include this gene. If the coin is another side, we should drop that genes. But how can we model that? That is our issue. So that is, we want to do some housekeeping for our genes. So we want to throw out all these kind of useless genes, but keep our some useful genes. So that we call that a spatial variable gene detection. That is a discriminator analysis. So that is genes are only active in specific automatic layers. So that are signal we actually want. So we want to throw out some genes cannot help us to discriminate this kind of spatial patterns. So that is, if we use all genes to the cluster, the noise downs out to the zero. So we need to automatically ignore some housekeeping this kind of gene. So that is our idea. So the idea, that is, we just have certain shift, or we just simply scientifically toss our coins. So that is our idea. So that is, we just have one switch on which gene should be included in our model. So that is, we have one this kind of switch. If the switch is on, so this gene is our, we call that discriminative genes. If this gene is this kind of off, we call this gene is housekeeping gene. So that is the idea. We have this kind of binary indicator to find which gene should be included in our models. And finally, what we can do, that is, we have another layer of this kind of two-way mixture. So that is our model. So we have, if this kind of gamma J equal to 1, so we include that gene. If gamma J not include 1, that equal to 0, they will have a non-discriminator analysis. So we will have this kind of baseline, this kind of stuff. So that is the idea. How can we select the genes in our studies? And next thing, that is, how can we find the clusters? So our cluster is from probabilistic models. So I just translate this kind of clustering process to some simple word. So we can think about if we have n observation, we want to cluster them to k clusters. So that is our goal. So how can we do that? We can treat that as n observation is n customer go to the restaurant. So if the customer sitting in the same table, like here, that means this kind of customer, this observation belong to the same cluster. So our clustering process that we can treat that is we assign people into the tables. The people sitting in the same table, we treat them in the same clusters. So now the issue that is, how can we assign their tables? We can think about this kind of clustering process sequentially. We call that is Chinese restaurant process. So that is, if the first people comes in, we randomly assign this person to one tables. He or she will assign one cluster. And then another people go into the restaurant. They have two choices, right? Sitting with the first people and open new tables. So that is, they have two choices. Other way, that is, if already we have 3 people or 4 people in this restaurant, 3 in the same table 1 and 1 in the table 2. So the customer 5 go to that restaurant, they have 3 choices. Go to table 1, go to table 2, or open a new tables. And what we want to do, that is, we just assign certain probability this customer going to which tables. So that is, how can we do that? And then what we can do, that is, we can sequentially to assign people in the table. Then these people automatically give certain clusters. So that is the idea. And the probability assign these people to one table that is proportional to the number of people in that tables. So that is the idea. So based on this kind of process, we will imagine this kind of stuff. So that is what we will have for this kind of studies. So, but in this kind of table, we ignore some very important thing, that is, if we go to a restaurant, we want to sit with our friends, right? We do not want to sit with some strange people. But this kind of process, this kind of assigned probability, just proportional to number of people in that tables. They do not quantify any relationship. So in our spatial context, they do not consider this kind of spatial correlations. So that is our idea. What we want to do, that is, we want to do some modification of this kind of traditional clustering process. So here, there are some 2 drawbacks of this kind of table. First, statistically, this kind of model cannot consistently estimate number of clusters. That is the first reason I like to adjust. The reason that is, if the true number of clusters equal to 4, this kind of method always will estimate your number of clusters go to 5 or 6. So that is the first reason. The second reason, that is this kind of geographical ignorance, since this method ignores the spatial maps. They do not quantify the relationship between the customers. So what we want to do is to solve these kind of 2 issues in this kind of clustering process. So our idea first, to fixing the cluster numbers, what we can do, that is, we just do some correction to, instead of using the DPMM model, we use the mixture of finite mixture model. So here, the mixture of finite mixture model statistically, okay, we just say that statistically, we can consistently estimate this kind of number of clusters. So that is, the estimation of number of clusters will be the true as your n goes to infinities. But mixture of finite mixture model still does not leverage this kind of geographical relationship. That does not quantify this kind of friendship. So what we want to do is move further. So our idea, that is, first, we want to translate our tissue to some spatial graph. So the global problem, that is, the tissue slice has 10,000 spots. Try to calculate how every single spot influence every else. That is very hard. It's very computation intensive. So that, for example, if you have 10,000 this kind of 10,000 spot, what you need to calculate is 10,000 times to 9,999 divided by 2, this kind of relationship. That is very hard. So our solution, that is, we just translate our physical tissue slice into some mathematical network graph. So that is, what we want to consider is one spot is neighborhood spot. So that is our idea. So that is, every sequence spot in the tissue becomes a single node, and there we draw certain lines containing this spot that are physically touched. So that is, we just consider neighborhood, and then what we want to do, that is, based on this kind of neighborhood, we build certain friendship. So that is our idea. So this idea, that is motivated by one thing called the Markov property, that is local dependence. So that is, if we want to consider the joint distribution, xi, given all rest of this kind of observation, what we want to do, that is, we just want to analyzing the xi with their neighborhoods, this kind of observation. So that is the idea. So that is the rule, that is, the sports cluster assignment depends only on its intimately touched neighborhoods and independent of the rest of the map. So that will reduce a lot of our computational this kind of burdens. So why this kind of make this kind of this method is perfect fit for this data. Biologically, cells only communicates via some physical contact and the local chemical gradients. So that is from our biological meaning. And also the analogy that is you are heavily influenced by the loud music playing in the apartment directly next door to you. Right? So that is some information what you can do, but you are not directly influenced by the house that is through 3 tons layers. So that is some idea what we do. So here, how can we construct in the statistically? So here, what we want to do that is we just add this kind of thing I call that is spatial peer pressure. So the mathematical info, the local dependence, we use a Markov random field. So that is based on the Markov properties. So we define this kind of cost or reward function based on this kind of neighborhood structures. So we add a certain reward to this kind of neighborhood structures. So that is this kind of identical matrix Zi equal to Zj, that is matchmakers, and this kind of neighborhood is our tally, and this kind of negative D is what we reward to this kind of neighborhood tally structure. So that is the idea how we do this kind of construction. So finally, what we want to do that is we can use this kind of D values to control our smoothness. So that this is one parameter we can control our spatial pressure here. So that is if D equal to 0, this model will reduce to traditional clustering process. Do not consider any this kind of spatial pressure. If D is moderate, we get this kind of biological flexible continuous tissue layers are smooth. If D is very high, the peer pressure is so intense, so the model will ignore the gene expression entirely, just for everything into one giant block clusters. So that is the idea. So this is our BMP algorithm automatically calculates the optimal D for use. So that is some idea, and our D can be learned directly from our data set. So that is the idea. This is a mathematical formula, so I would like to ignore that. But the idea that is, if we ignore this kind of red part, this model reduced to our traditional mixture finite mixture model. But what we do that is we can add this kind of spatial boundless part to increase for existing table this kind of friendship. So you can see the traditional model still just depends on this kind of number of observation in that table. But here we add this exponential part, since we know that exponentials one positive value that will be greater than 1. So the probability for existing tables, that probability will be the higher than the traditional. So that is our statistical formula. Now the next thing that is how can we do the Bayesian inference? So if we have a model, then what we need to do that is how can we get an estimate for all this model? So idea that is doing this kind of collapse skip sample algorithm. The beauty of this kind of algorithm that is a lot of traditional algorithm, we need to first to know the K, number of cluster, and then assign observation to each cluster labels. But our idea that is we ignore this kind of K, we collapse this K. What we want to do that is we just to randomly sample all these kind of clustering assignment for each observation. What we need to do, just need to summarize the distinction number in that cluster assignment, then we know the K. So that is the beauty, how can we have this kind of Bayesian inference. And then another thing that is, we also need to inference which gene is very important. So what we can do that is we just have this kind of posterior probability inclusion stuff for each gene. So that is, we calculate this kind of probability, summarize all the sample probability, this and gamma j equal to 1 given our data, and then since we have a bunch of genes, we need to do some forced discovery FDR control. So that is, we will do the FDR control to select the final list of spatial variable gene. And the final thing that is, very important thing that is, when we have this kind of cluster assignment, that is, we use this kind of co-clustering matrix to summarize our clustering assignment for our observation. Okay, as time limit, so I show this kind of simulation studies. So this is some true spatial cluster we have. We have 3 different patterns. We want to see how our method is truly recover this kind of assignment. So here, this first, that is called just random index. So we can see, we compare our method to a lot of benchmark method. This random index is from 0 and 1. 1 means that you have perfectly matched our true cluster. 0 means that it is randomly guessed. So we will find that our method, the first is our method. We can see our method is very close to 1 over replicates. And also we can see how our method truly recovered the number of clusters in our settings. So here we can see our sensitivity, specificity, this kind of miscalculating, this kind of clustering rate and AUC is very high in all this kind of setting based on different this kind of dropping rate. And this is from this kind of spatial platform, how can we generate our data set as a result. This is our final result. So for a lot of this kind of tissue, a lot of doctors know what is a true spatial cluster here. So that is, we have our ground truth, and we want to compare our method with some other method based on this ARI in the real data analysis. We can see that our ARI is much higher than all these kind of computing methods in this kind of first data set. And this is our discriminant genes we select from the original maybe 20,000 genes we have. And this is another data set that we also can see our method have. You can see, visualize, we can see our method have better this kind of clustering result than all these kind of benchmark methods. And also this is identifying this kind of spatially variable gene based on our study. We have this kind of 6 key discriminant genes automatically identified by our internal feature selection methods. And this is some theoretical substructure we can detect from our methods. Okay, so that is all we have. Give a conclusion that today my talk give you a unified framework that joins technical clustering and feature selection in one single elegant Bayesian step. So that is something I want to do. And also, this method is high fidelity. By operating this kind of totally original count, we pervert the biological signal noise associated with the dimension reduction. That is what we want to do. We do not want to lose too many information based on our pre-processing. And also, all these methods are fully automatically. You do not need to specify K. You don't need to specify how many genes you want to include in your model. Our model can automatically give you how many genes you should include, what kind of K you need to have, so no more guessing. And also our method is spatial aware. We have this kind of spatial pressure or boundness in our model to encourage nearby genes will be spatial clusters. And also compare with some benchmark method, our model have this kind of superior result. Okay, and finally, I will send to my collaborator Aura Reward. The Ben Chong is the PhD student from City University of Hong Kong, and Xiaodan is his advisor, and my medical collaborator from the University of Texas South Medical Center, and my long-term collaborator from the University of Texas at Dallas. That's all today. Finally, I want to give some final remarks from our models. That is famous quote, not all the models are wrong. So the original one is not all models are right. But my quote is, not all models are wrong. In fact, some of them could be correct, at least locally. From a statistician, what we want to do that is, we can extract high useful biological information from them. Thank you.
All righty, thank you, Guanyu. We have time for just a couple of questions. Raise your hand. Oh, we've got a question right here right in front of us.
Yeah, thank you for the amazing talk. I mean, like I mentioned, I'm really interested in like data science and stuff. And I was just wondering that with the models that you talked about with the Bayesian models and things, have you guys compared the results that would be generated by these distributions to real genomics expression data?
Yes. That's a good question. And that is, I just ignore that part. And that is, the first simulation we did is for this kind of mimic this kind of spatial structure. But here, there are another simulation that is on the real this kind of spatial omics data platform that we call the Visium data platform. So this platform is this kind of platform structures. We also generate that. And also we see that is that if you ignore this kind of column data, that will lose a lot of information when you correctly specify the number of clusters. Yeah.
Thank you.
I've got one here. Quick question for you, and that is just if you could help me appreciate the magnitude of the problem. You talked early on that you could have your genes could be given a 0 or 1 depending on whether or not you see anything. It could be a real one or a false one. Yeah. If the methods weren't collecting the gene data for whatever reason. To what extent what percentage of the zeros are real zeros versus false zeros? And is that number falling precipitously as the techniques to isolate the RNA improving over time?
Yeah, so that is a good question. And that is, since here in our models, we have these kind of binary indicators. So here we assume that's our assumptions. So here, this kind of pi is this kind of proportion, how this is come from this kind of technical issue zeros. So that is what we did. So based on this kind of stuff, and also in the simulation study, we generate different kind of zero proportion. So here, this is the dropout rate. Yeah, this kind of is dropout rate, and also we also see that is how our model accurate to estimate this kind of dropout rate, and also in our simulation study we can validate our method can correctly estimate this kind of dropout rate. But in the real data, definitely we do not know. But this based on our this kind of assumption that is our this kind of zero comes from two parts. The true biological zero comes from the Poisson part, but this kind of technical zero is come from this kind of inflated part. That is our assumption. Yeah.
All right, thank you. All righty, I think we are out of time, so I just want to say one thing. I really appreciate your comment about the difference between correlation and causation. As all scientists know, a correlation does not necessarily equal causation. Yeah. So thank you for that, for reminding us that. So let's all thank Guanyu again.
Thank you so much to all of our wonderful presenters, everyone still listening online, everyone here, and all the great questions being asked. We appreciate the engagement and the excitement immensely. So we are going to go into a break to have some snacks, regroup, and talk about what we've learned so far. So we'll take a 30-minute break, and we'll start back at 11:36. So 30-minute break, and we'll be back at 11:36. Thank you.
All righty, welcome back. And I hope you all enjoyed having the opportunity to talk with one another and to get even more of your questions answered about the talks we heard earlier today. I know I jumped in and got some questions answered as well. So welcome back, everyone. And those of you joining us online, again, also welcome back. We hope everyone had a chance to get something to eat because we are ready to rock and roll on two more really exciting talks. Next, I would like to introduce you to Suhaylah Ahmad Ali. She is a senior zoology major and a Dean's Research Scholar who will tell us a little bit more about the work that she has been doing here on campus. So please join me in welcoming Suhaylah to the stage.
Hello, everyone. Okay. Thank you for everyone for waking up so early for this. I hope you love waking up just as early as I like to wake up. So my name is Suhaylah, and today I want to introduce you to wildlife disease ecology. Basically the study of how wildlife, parasites, and the environment interact. And specifically, I want to talk about 3 principles or 3 common themes across 3 projects that I've worked on in my undergrad. But first, a bit of an introduction. Again, I'm Suhaylah. I am originally from Pittsburgh, Pennsylvania, so I'm an out-of-state student. I have a major in zoology with a minor in environmental and sustainability studies. So I really love the environment. I currently work in 2 research labs, and my primary interest is wildlife disease, parasites, wildlife, environment. And fun fact, I am an artist, so you'll see some of the art that I've been able to work on in my undergrad as well, and really been able to integrate into my research. Let's first get into the first principle. And so that first principle I've learned across this particular project is that first, there's a lot of hidden complexity in disease systems. So if we're looking at this, let's say particular parasite I've studied called T. bryosomane. So this is a microscopic parasite, and it has a host. Its particular host is salmon, any species of the salmon family. So coho, king trout, rainbow trout. And so in wildlife disease systems, we're commonly used to thinking about the parasite in the life cycle and the host which it impacts. So T. bryosomane is deadly to salmon. The spores are in the water, they enter through the gills, they reach the kidney, and that can be fatal to the fish. But while studying this parasite, I realized that is not actually the case. It ends up becoming much more complex, and the life cycle of this parasite actually also includes a first host or intermediate host called bryozoans. So bryozoans you can kind of think of as like freshwater coral. They kind of are sedentary, and then they grow by releasing their spores and reattaching to rocks in freshwater systems. And they do the same. They take up T. bryosalmone, and then they kind of help transmit the parasite to salmon. And by the way, bryozoans, they exist like right along the Red Cedar River here in campus as well. And so I was studying this parasite, and I was interested in knowing what other filter feeders are able to take up the same spores. So I looked at invasive mussels, and we have a lot of invasive mussels, zebra mussels, quagga mussels here in Michigan. And so I wanted to know, are they a suitable host? They act pretty much like bryozoans do, and they filter the water to get food. So I got scholarships to do an independent project, and I sampled like about 300 to 500 of them and processed a bunch. And based on however many positive samples I found, I realized they probably were not. So we only found like 4 positive samples in 300 mussels that I dissected and ran PCR for and ran sequencing for. So like 6 months of work, and I was like, oh, that's not significant. There's like a 1% prevalence. But it is novel. No one's ever tested invasive mussels, which are everywhere in Michigan, for this particular parasite. So it's incredibly interesting, and again, it was like a sample size of like 300, but mussels, invasive mussels specifically, grow in the thousands. So that definitely adds complexity, and it's new. We did not know that the DNA of this parasite could end up in the tissue of these mussels. So that got me interested in other systems. And that brings me to the second theme or principle, and that's that environment shapes parasite diversity and the different kinds of animals that the specific parasites interact with. So the next parasite that kind of jumped into, because I can't just work with one, I needed to work with more. So the next project that I jumped into, which I'm currently working on, is a project on saprolegnia. So instead of a parasite that infects fish kidneys, they grow more like a fungus. So I was interested in culturing and discovering what kind of saprolegnia strains could infect fish. And they infect fish and kind of grow like a white cottony fungus, and they're really widespread. But their species are not well described here in Michigan whatsoever. So I ended up being able to collaborate with researchers in Budapest, Hungary, who are studying where it is most prevalent, and they're interested in why it's a big problem in fish hatcheries. So as an example, you'll see here's a photo of a salmon that is very heavily infected with this fungus-like parasite. And so I did the same thing. I took water samples, mucus samples, and I put them on culture plates and see if I could collect any strains here, because there's not any information on strains that exist here in Michigan. So I put them on culture plates, and I actually grew some. I did not expect to do that. I actually got a particular less prevalent species of saprolegnia called Saprolegnia australis. And not only that, we discovered 2 lineages right here in Michigan in the water, including Indiana. So we had 9 total isolates, 2 separate lineages. In red, they're from Bodine, Indiana, and that branches off from the first group in blue, which is from Platte River, Michigan. And so why this is interesting is because early experiments in Hungary by those researchers I'm working with who have taught me everything about the life cycle of this parasite is that the lineage in red has actually been shown to be warm tolerant, and the lineage in blue has been shown to be cold tolerant. This means that they're so different, in fact, that one lineage is actually more used to extreme high heat temperatures, and they grow best in a higher temperature. And one of them, which actually is from further up north because it's Platte River, is actually used to growing in much more cold extremes. And so this tells you basically that they definitely evolved in they're used to different environments, and because like as the environment shifts and the temperature extremes we experience are pushing them to opposite extreme ends. But again, it was like nine isolates that I have. We have more samples that are growing in the lab, and we're going to be able to push this experiment further into the summer because we collected these in cold weather. So we want to see what kind of lineages we get later in the summer. But this all couldn't have been done without the start of my research, and that was through the fish pathology and immunology lab. We are at the food safety and toxicology. So my PI, we study fish parasites, viruses, freshwater organisms, invertebrates, and aquatic ecology. Could not have done it without them. And all of this starts with fieldwork. So I have been all over Michigan, in the river systems, in fish hatcheries. We dissected all kinds of fish, and if you think molecular work is a lot of work, field work is chaos. So that's been incredibly super fun, and we'll be continuing culturing pathogens and studying aquatic ecology and aquatic parasites and how deadly they could be to fish, how much of a problem they could be with changing climates later as time goes on. So just a hard pivot, actually, because I was interested in parasite diversity. I also started gaining interest in land use because we studied parasites in hatcheries and aquatic systems, but they all run through different kinds of land, natural land, agricultural land, through farms. So through another lab, the second lab, I was interested in this third principle, it's just the fact that land use influences host-parasite interactions. So big switch from fish, now we're looking at birds. Specifically things like avian malaria and parasites that can cause avian malaria. So in this second lab, I was interested in sampling blood from birds across natural and agricultural sites. And so in addition to that, we caught a bunch of birds. We were interested in looking at blood parasites like avian malaria, as well as how that relates to their body condition, the glucose we measured, the ketones, and the white blood cell counts. And we smeared all the blood on slides and spent hundreds of hours just looking at the blood and seeing, okay, what can we find? How does this impact the birds that we caught? So just a subset of that data was black-capped chickadees. In total, we caught over 400 birds. But yeah, so this is just a subset of 66 black-capped chickadees. You can see them all over campus, and they're all from natural sites or wooded areas. Toomey woodlot, Baker woodlot, they're all near campus here. And so we wanted to know, okay, how does the body condition that we measured, how are they affected by parasites, the parasites we found, and how does the landscape, the woodlots that we found them in, affect that body condition? Just a quick summary of the kinds of parasites we found. So out of 66 black-capped chickadees, 12.1% of them had trypanosomes, which are not avian malaria, but they kind of look like a worm, but they can be deadly, and we did not expect to find them in black-capped chickadees. And 9.1 of them had hemosporidian or malarial parasites, which can cause avian malaria. So at the top are trypanosomes, and the bottom are the two avian malarial parasites. And then there was a ton of statistical analyses that I did not expect. But basically, again, we wanted to know how the landscape or conditions affected body conditions, and if any of those measurements were predictors of body conditions. Out of all of them, we didn't find any significance of all the locations compared, of Baker woodlot, Toomey. We've also looked at a residential area. So yeah, not significant if we grouped all the locations together and said, okay, how does the number of parasites or these measurements we took like glucose and ketones affect body condition? Not significant. But there were individual locations that show strong relationships, such as in the bottom squared graph, you'll see that hatch year, which are the younger birds as opposed to adult birds, the hatch year individuals had reduced body condition at inland lakes. Inland lakes has a bunch of surrounding bodies of water. So those younger birds there had reduced body condition, but they were not significant at other locations. And there were other individual significance at specific locations. But this basically tells us is that there's so much variability across all the landscapes. There's significance within specific landscapes, But if you group them all together, you might not see anything significant, and you might be like, it's not worth taking blood from these birds anyway. But each of those landscapes are actually quite far apart from one another, and there's very specific, significant differences we can find in those individual places. And I did all this through the McGraw lab, which is sort of the continuation of my research. So in addition to fish lab, I'll be doing more field sampling with my bird lab. So this is through the College of Natural Science specifically. And that photo there is a drawing actually I made for the bird lab logo that we'll be wearing during the summer. So if you'll be wondering where I am, just look for a bunch of undergrads with that on the back of their t-shirts huddling around, either like hauling mist nets or like, wrestling birds. So yeah, that's where we'll be this summer. But the moral of the story really is that, in a short summary, is that fish and birds and many other animals carry many complex parasites, ones that I certainly didn't know exist. The diversity and severity of the impact of these parasites vary widely across landscapes due to land use, resource variability, and wildlife diversity. It's important to keep track of these changes because in parasite animal and in ecosystem interactions, because humans do inhabit these spaces. Again, we've sampled at residential areas, so people live around these spaces, and so we are a part of the system. And if I've learned anything, it's that any negative, like at any disease outbreak that there is, either whether it be in humans or animals, there often is because of a stressor. And stressors happen when there is a pressure on the system or the animal. And because we're part of that system, we need to be careful of either the stressor we put, the impact we may have, or what stressors may have a bad outcome on humans. So that's sort of how those all tie together. It was a bit rushed, but thank you for listening to it. I'm happy to take any questions about it.
All righty. Thank you, Suhaylah. We have time for about 2 questions. I see one right there.
Thank you. That was awesome. So when you talk about stressors and things to limit these parasites in these fish and bird species, are there any specifically that come to mind to you that you have found that have made a significant impact? And if so, how can we as people kind of help these non-human species out?
Yeah, so when I think about stressors, something that immediately comes to mind to me is pollution. As I showed in the graph before, so here we found a couple of significant predictors of different measurements we took from birds on body condition at places like inland lakes. Inland lakes is the only place that we studied at that has large bodies of water surrounding it. And something that water is prone to being affected by is pollution or excess nutrients, runoff, something like that. And so that is a very common trend that we'll see, is that waters that are polluted or waters with tons of nutrients and tons of wildlife flocking to one area, that creates a stressor because there's competition around the areas with either lots of resources, or if there's tons of pollution, that pollution can then impact the resources that animals end up consuming for energy. So that can end up being a stressor, and any stressor that reduces an animal's immune system will then leave them susceptible to infection.
We have a question from one of our online participants, and that question is, how do you collect the birds to study the parasites?
I briefly mentioned, so I wish I had a photo of it because I wasn't available for fieldwork the previous summer, but I will be this summer. We just haul around giant mist nets, and we work with local farmers, and we work with the Michigan DNR to be able to basically reserve certain sites near trees where we can set up giant mist nets. And so those are like nets that you can barely see through, but the birds will fly in through those nets when they're not being bothered and they are not looking. And basically everyone is like, jumps on the bird. Everyone runs to catch the bird. And we take the blood through like an artery on their wing. So we'll take blood samples from each side of their wing. We'll band them so we can remember them if we ever catch them again. And then we'll let them go. So it's non-lethal. It's a really good way of doing research and collecting valuable data without actually harming the populations.
All righty. Thank you, Suhaylah. And I just want to say one thing quickly. Thank you for your message about the stressors and the impact that humans are having on the stressors. So that's good for all of us to remember. So thank you. Let's thank her for a wonderful talk. All righty. So our final presentation today features Dr. Sean Crosson, the Rudolph Hugh Endowed Professor in the Department of Microbiology, Genetics, and Immunology. The Crosson Lab focuses on the biology of bacteria that inhabit the environment, all the way from fresh water to soil to the interior of mammalian cells. Now, when we think about microbial life, many people sort of default to thinking about infections and disease, and of course that's really important. But microbes also play an essential role in supporting animal and human health. And of course, we all know about the gut microbiota, for instance, and things like that. So we know how important it is. But they're also involved in sustaining the soils and plants that underpin our soils, our food systems. So today, Sean will highlight how his work at MSU spans this One Health continuum that we talked about earlier today, from host-pathogen interactions in bacteria that cause disease in livestock and humans, to the environmental bacteria that support human health and that support soil health and plant resilience. So together, these explanations show how microbiology together connects human outcomes across hosts and ecosystems. And this truly is one of my passions, so I can't wait to hear. So let us please welcome Sean to the stage. Let me just pull this up. I think this is me. Come here. Okay, that's me. Great. All right, cool. Thanks. All right. Thanks, Dean Hegg, and thanks for the opportunity to speak to you all today. I just want to start off by saying that I mean, science is it's not one dude in the lab toiling away by himself. It's a team endeavor, and it's really through the intellectual generosity of all of my collaborators that I have the privilege of standing up here and telling you about the work that we do. We collaborate with a lot of people here at MSU. It's a fantastic collaborative environment, but also with different groups around the world. And I want to start off with a quote from Rudolf Virchow, who's considered the father of modern pathology. Between animal and human medicine, there are no dividing, and this is translated from the German. It's a series of famous lectures that he gave in Berlin in the 1850s. There are no dividing lines, nor should there be. And this is really, this quote is at the foundation, or is one of the quotes at the foundation of the modern One Health, One Medicine concept. And he came to this through inspection of diseased tissue under the microscope, human tissue and animal tissue, and realizing that the cellular defects were the same. Virchow also coined the term zoonosis. These are diseases that are transmitted from animals to humans. Of course, he wasn't, if you read Leviticus, right, you know that there was some human concept of animal to human transmission, but I think Virchow put this on a firm footing scientifically as the germ theory of disease was really sort of crystallizing, even though Virchow rejected the germ theory of disease. Okay, so I like this quote, and I just here we have the modern incarnation of this One Health framework, and just are there. I was just seeing if y'all could see my cursor. You can't, so I'm just going to talk and point. All right. So like Dr. Hegg's talked about, there's this cool Venn diagram where human health impacts animal health
impacts plant health. They're all connected. And what I want to tell you today is that microbes are really the thread that sort of ties all these circles together. Microbes touch everything. And I want to just give you a few examples. The first little story I'm going to tell you all about is about this bacterium called Brucella, which is a massive animal health problem globally. And a number of Brucella species are zoonotic pathogens, and they infect human health. They impact human health. I also want to tell you about this bacterium called Caulibacter, and this is a Caulibacter species that was pulled out of the dirt, pulled out of a soybean field down at Kellogg Biological Station, and it actually seems to support soybean health, right? And then I want to tell you all about this bacterium that's a common resident of our gut, Bacteroides fragilis. It's there and it's doing its job, but under certain disease conditions, inflammatory bowel disease, this bacterium can bloom. It sort of explodes in numbers, and that's associated with gut inflammation and disease. All right, so I'm going to start with brucellosis. I grew up in a rural area of Texas. This disease was a major problem in the 70s and 80s when I was growing up. It's also called Bang's disease. I don't know if there's any sort of bovine folks here. But it's really a neglected disease. There's millions of animals infected, livestock and wildlife worldwide, and an estimated 2.1 million human cases a year. And that's almost certainly an underestimate. People who have brucellosis are often misdiagnosed with malaria and other diseases, and they're not treated correctly in the clinic. It causes significant agricultural losses, sterility and abortion in livestock, and this is the ag impact. And there's a lot of species of Brucella recognized now that infect a lot of different wildlife, animals, and livestock. The ones that are best studied are these livestock pathogens. You can see here melitensis in goats, suis in pigs, abortus in cows. And their zoonotic potential, their potential to infect humans, is indicated by these red arrows. So some species are zoonotic, some are not. And it's a sneaky bacterium. It's cool. It infects us, normally through our mucosal crosses a tissue barrier. And then it invades. So the Brucella are cartoonified here as these little red dots, okay? And the big cell, and so this gives you a sense of scale, okay? They're really small because this big cell is it's a macrophage. It's just like a flavor of white blood cell, okay? And it invades macrophages. And normally the job of the macrophage is to engulf foreign invaders and kill these, right? But a fraction of Brucella are able to sort of trick the macrophage, this immune cell, into trafficking it into a little compartment called a Brucella-containing vacuole, or a BCV, and that's where its home is, and that's where it replicates. So it makes its home inside cells that are supposed to kill it, right? So it's sneaky. And then from there, during the chronic phase of infection, it can spread from cell to cell. And we're really interested in understanding the genes that are required for Brucella to infect these mammalian host cells, human host cells. We're also getting macrophages from the dairy, collaborating with veterinarians here at the CVM, collaborating with immunologists in our department, Andrew Olive. This is really a big collaborative project. And so as we think about genes that are required by Brucella has about 3,000 genes, right? And we want to know, well, what are the genes that are required for it to infect these immune cells? And so we do a lot of genetics in my group. And the approach it's kind of a funny approach. We use these little pieces of DNA called transposons that randomly will jump or insert into the chromosome. And we study things by breaking them, right? Where a piece of DNA, a transposon jumps in, it inserts into a gene and it breaks it. And we can do this for thousands of genes. And in one shot, we can interrogate, what are these genes? And the strains where the gene's disrupted, we call those mutants. Okay, and we can ask, what are the mutants that are no longer able to infect the host cell, right? And so using these approaches where we go in and just bash the genome and break things, and on the backside, we can sort things out using modern DNA sequencing approaches. We found genes that when they're disrupted in Brucella can no longer enter the host cell. And we found genes that they get in just fine, but then they get killed, okay? So we're finding these pressure points genetically in Brucella, that genes that are required for infection. Now, of course, now y'all can extrapolate from there and you can say, well, okay, well, those are potential targets, right? If you could go after those genes, if you know those genes and you know those processes, those are things that could be targeted therapeutically. All right. But we're not only looking at genes that are required for the bacterium to infect the host, and this is sort of a strange concept. Maybe it's not a strange concept. I mean, y'all can imagine, like, here's this, let's say that's a human macrophage, and we do work on human macrophages. There are processes and pathways in that host cell that are required for the bacterium to infect that host cell. Okay? So if those processes are messed up in the macrophage, then the bacterium can no longer get in. And we would like to understand those better. And in collaborating with Andrew Olive's group, using modern CRISPR-based genetic approaches, we are making mutants, we're mutagenizing, we're bashing the genome of these macrophages. And we have found genes that, when they're knocked out, Brucella can no longer get in. And we found genes that when they're knocked out in the human macrophage, Brucella gets in, but it gets killed, okay? So these are host factors that support infection by the pathogen, right? And an interesting concept here is like, well, you know, now that we know those pathways, a lot of these pathways are really well known and they're understood, and there's actually drugs that target those pathways. And so the question is, and this is a collaboration, a screen that we did with Rick Newbig's group here at MSU, well, can you drug the host to make it less hospitable to the pathogen, right? And this is called host targeting, host-targeted therapeutics. And one potential benefit of this approach is normally when we take antibiotics, right, you're like, you know, going scorched earth on the bacterium, on the microbe. Here you're treating the host to make it less susceptible to the microbe. And we've actually found drugs, and some of these are actually FDA approved, that when we treat in a dish, it's the same sort of deal. We can treat with these molecules and it makes the host less susceptible to entry, or it makes the host better able to kill the bacterium once it's inside. So, you know, I'm not showing y'all much data here. I'm just talking like concept. But if anybody wants to see data, I'm a scientist. We'll be like, well, what? I don't believe you. I'm happy to sit down with y'all and look at the data. All right, but I just wanted to give you some conceptual idea how we're thinking about this. Okay, so, you know, Brucella, it's this bad agricultural and human pathogen, right? And so this is the tree of life, and y'all may have seen these. This is an abstraction, right, based on a likelihood model of alignment of a bunch of DNA sequences of the diversity of life on the planet, all right? And y'all have seen, I'm sure you've seen these trees before. This goes back to like Darwin, right? The eukarya there on the far right, y'all know where we sit on this tree? We're in there with plants and fungi and protists. In terms of diversity, we're pretty minor of diversity of life on this planet. Just to the left of the eukarya, those are the archaea, really interesting microbial cells. A lot of them look like bacteria, but they are absolutely not bacteria. And then to the far left, that's the bacterial world, okay? And you can see the alpha proteobacteria down there. It's like near, I'm gonna put a little star in here. That's where Brucella sits. And Brucella has a cousin, Caulobacter. Taxonomically, I don't know if that's kosher to call them cousins. They're really closely related. They're on that same branch, okay? Caulobacter is a different beast. It lives in soil. It lives in fresh water. It's not pathogenic, right? Unless you're severely immune compromised. There's some crazy clinical cases of Caulobacter infections. This is strain, and this is an electron micrograph that was taken by collaborator Aretha Feebig and our joint student, Maddie Quinlan, here at MSU. And you can see there are these beautiful little curved cells, and there's those threads. That's sort of sugar that it secretes, these sugar strings. And this was isolated by an MSU student in our department, Reed Longley, from a no-till soybean plot down at Kellogg Biological Station down by Kalamazoo. All right, and so why was Reed doing this work? Well, Reed was interested in the bacteria that are associated with soybean roots. And so have y'all seen soybean roots before? They're interesting. They have these little nodules on them. Can y'all see those little bumps, those little circles? Inside those are bacteria known as Bradyrhizobium, okay? But it turns out there's other bacteria in there too. And I'll get to that in a sec. But what do those nodules do? Well, nitrogen is an element that's required for life. We're bathed in nitrogen, right? 80% of what we're breathing right now in and out of our lungs is nitrogen, but it's in this N triple bond N form, super stable molecule, not biologically usable, right? But, and what we do to make fertilizer is we burn boatloads of fossil fuel, right? It takes a lot of energy, and we make ammonium, NH3, ammonia, NH3. And that's a usable form of nitrogen, okay? But these bacteria that live in these root nodules, they can just do that. They do it for free. They take nitrogen out of the air and they make fertilizer, right? They make usable nitrogen, so that's cool. And the presence of those nodules and the presence of those bacteria inside the soybean root is really important for plant health and plant production, right? And so Reed was looking at the other bacteria that are present in the soybean root, and he pulled out this Caulobacter, right? And we're like, okay, well, that's super interesting. What is it doing there? And what Reed did is he took the dirt out of this no-till field from Kellogg, brought it back to the greenhouses here, planted a bunch of soybeans, and then added live Caulobacter cultures, sort of watered them with these bacteria that he pulled out of the root, and that they made more nodules. And they had more of this good bacterium in their roots that's giving them nitrogen, that's fertilizing them. So it seemed like a really good thing. And so then he did another experiment where he subjected these plants to drought stress in the greenhouse. And he took either no bacteria added, and on the y-axis here, the vertical axis, we're just looking at plant weight, okay? And you see no bacteria, under drought conditions, 160, MIGs. Heat killed bacteria. He killed the bacteria and added it. Didn't make a difference. But if he added live Caulobacter, the plants did better. They grew better under this drought stress. Right? So it's kind of like a plant probiotic, right? And so I love that result, right? Can we just add more bacteria to a field that's already there anyway to make things better, right? And so we, wanted to well, how does that work? And there's a lot, you come up with ideas, you come up with hypotheses, right? And so this is a collaboration with my longtime collaborator, Aretha Fiebig here at MSU. And so I told y'all that these bradyrhizobium in the roots, those are a good thing, right? They're giving the plant nitrogen from the air, right? And there's this Caulobacter that also seemed to support plant growth under drought conditions. Well, one possibility is that the Caulobacter is making the plant do better directly. Another possibility is that Caulobacter is helping out this other bacterium. It's a helper, right? And it helps the bacterium that gives the plant nitrogen. And so we tested this, so just directly. So we made dirt juice, and we poured some Petri plates with growth medium, and we plated Caulobacter, Caulo, and bradyrhizobium, Brady, at different distances from each other on these Petri plates with soil extract and a different carbon source. And we just assessed growth. And what you can see, actually, I love this. When bradyrhizobium, this is the bacterium that's given the plant nitrogen, is close to Caulobacter, it grows. And the further it gets away, it doesn't grow as well. And this is true on these are two 5-carbon sugars that are relevant in soil. So it really seems like on plates, Caulobacter is supporting this bacterium, right? And that might explain what's going on in the plant situation. And I think what's even more interesting is we're looking at increasing temperatures here. So 37 degrees, right? That's body temperature, right? Soils hit 37 degrees commonly. And you can see that bradyrhizobium, the one that's given the plant nitrogen, it really wilts as things get warm. But when it's close to Caulobacter, it does good, right? So Caulobacter helps it out under stress. And so the way that we're thinking about this bacterium, which is a close cousin of this really bad agricultural pathogen, is it lives in the soil and it's a helper. Its role in soil is to support other microbes. And particularly, it seems like there's evidence that it supports this other bacterium that makes its home inside soybean roots and really helps crop production, soybean production, right? And of course, soy is a really important crop here in Michigan and around the world. So that's our model now, and we're continuing to push on that. I want to close with a story about inflammatory bowel disease. And this is a collaboration with a group of gastroenterologists at the University of Chicago led by Jean Chang. Mitch Sogan is a microbial ecologist at the Marine Biological Lab in Woods Hole. Marat Aaron is an ecologist at the Helmholtz Institute in Germany, and Aretha Fiebig here at MSU is also working on this. And there's a lot of, flavors of inflammatory bowel disease. What we're talking about here is ulcerative colitis. This is inflammation of the colon. And IBD is really complicated disease. It's multifactorial. There's a host genetic component. There's an environmental component. There's a gut microbial component, right? And we know that because these patients are often treated with antibiotics and they get better after antibiotic treatment, right? And there's 3 million people in the US living with IBD, and that number has gone up. This is a disease that's increased in incidence dramatically over the past century, right? And it's not because clinicians in the 20s didn't know how to, diagnose an inflamed colon. They did, right? There's almost certainly an environmental component to the rise of this disease. It's probably diet is a contributor, almost certainly, right? And so one of the things we know, I'm a microbiologist. I'm going to tell you about I'm going to tell you a microbial story. One of the things that happens in these patients when they get gut inflammation is like normally you have a balance of microbes in your gut, okay? But under conditions of inflammation, there's certain species that just bloom, they explode. And this really leads to an imbalance of the population of microbes in the gut. And one of these is Bacteroides fragilis. It's a normal resident of the gut, about 1% ballpark. But in certain patients under inflamed conditions, it can become 70% or more of the microbes of that population that's living in your gut. And so, we're interested in really a fundamental question, like why do these blooms occur? What's underneath that? And it's a question I'm not going to answer today. This is really a long-term question that a lot of people in the world are thinking about. Do these blooms contribute to inflammatory bowel disease, or are they just simply a consequence of inflammation? Are these gut microbes that just do really well when the gut becomes inflamed, right? And they have nothing to do with disease. This is a correlation causation thing, right? All right, so I'm just going to tell you about, you have an inflamed colon. There's a lot of things that are different physically and chemically about that environment. One of these, I'm just going to tell you a couple. One of these is that the level of bile acids shifts dramatically, right? And so we all know bile, comes from the gallbladder, secreted into the gut, assists with emulsification of fats and digestion. These bile acids, these like sterile-looking molecules on the left, they have important roles not only in aiding digestion and breakdown of food, they have important signaling and regulatory roles in the gut. And they're also toxic to bacteria at certain levels, right? They have detergent-like properties, and they're harmful to bacteria once you get above a certain threshold. Ida Heptesian's group at Stanford has measured the levels of secondary bile acids. And here I'm just showing you a control patient on the left, and we're looking in particular at deoxycholic acid, which is the molecule on the left. You can see that's normal levels in a control patient. In an ulcerative colitis patient, the levels of secondary bile acids go way down. And so you can imagine this is a situation where you have this molecule that's normally around sort of putting the brakes on microbes, and when that becomes low, when that goes down, now the microbe is able to bloom, right? That's one hypothesis. Okay. Another thing that's really dramatically different in the guts of patients with inflammation is the level of sodium, right? There are normal transport processes that lead to absorption of sodium and water in a healthy colon. And these are really disrupted in patients with ulcerative colitis. And so you end up with really high levels of sodium, right? High levels of water. That's the reason these patients have really watery stool, right? And so you have lower secondary bile acids and higher sodium. And so, these are just two chemical conditions that we're thinking about as we think about these blooms. And to study these blooms here, and I'm just going to summarize things really quick, our approach, we have patients that are in the clinic with ulcerative colitis, and we've isolated Bacteroides fragilis from these human patients. Patient 207 in particular, we have a Bacteroides fragilis from a bloom, and it's the same game that we play with Brucella, right? We go in and we bash the genome. We start breaking genes, and we ask, what are the genes that are required for these microbes to grow or thrive under conditions in the laboratory where we're trying to simulate what's going on in the human gut, right? Elevated sodium, lower secondary bile acids. And the bottom line is, as we're discovering genes and processes, it seems like lower levels of secondary bile acids and higher levels of sodium. It seems like jointly Bacteroides fragilis is able to leverage these two things, and that really assists its growth in the gut. All right. So I hope that I've showed y'all just a little bit here about what we're thinking about with respect to the connection between microbes and animal health and human health and plant health. And our hope is that the fundamental studies that we're doing are going to inform better treatments of microbial infection. But we also hope that the work that we're doing are going to maybe lead to better solutions to support plant health and agriculture. I think we think about microbes, I don't know, I think about my, folks I know, microbes are all bad. They're not all bad. They do good things too, right? And I think there's hopefully microbial solutions to a lot of our global problems down the road, and I hope that we can contribute to that. And I'm happy to take any questions. Of course, I just want to say, look, like I said at the beginning, I'm just up here delivering, saying words. It's a team effort, and graduate students and postdocs in the lab and our collaborators are really huge. And of course, funding sources, without them, we can't do any of this. So thanks for your time.
All righty. Thank you, Sean, for really a fantastic talk. And I really appreciate the way you kind of brought the microbes in, sort of the central of this One Health. And I fully embrace the way you think about it. So in any event, we've got time for about 3 questions. We've got one right over here.
Yeah, I'm going to kind of indirectly ask you for a little bit of investment advice, I think. I hear your talk, and if I would have invested in hand sanitizer in 2019, I'd be rich by now. And I'm sure you go through a lot of hand sanitizer with your work. But fertilizer is on the precipice of being on a shortage. Yeah. And what do you think that's going to do to the microbes and the plants and the farming over the next year as we have the Strait of Hormuz currently closed for about a month and counting, and that's going to shorten the fertilizer supply?
Yeah, this is a huge problem globally, right? It's obvious that the price of fertilizer is directly tied to the price of oil, right? And this is impacting farmers everywhere, right? And so I really love this example, and this is a soybean specific example here, but I think there are microbial solutions for soils to support plant growth, right? If you could figure out some way to not have to buy fertilizer and you could just put microbes in the soil, if you could supplement the soil in a way where they're doing that for free, right? They're taking nitrogen out of the sky and making it making it ammonia, right? And so I think these bioinoculants, this is what people call them, and we're not the only people thinking about this. There's other folks here at MSU. Sarah Liebes's group is thinking a lot about this with soy. I think people need to be thinking about microbes. People need to be working on these problems, right? Because if we can, and I think it's going to take a precision thing. A soil from the thumb is not the same as a soil from my home in Texas, right? But if you can figure out what the microbes are in that soil and what's working together, right? I think maybe we can come up with sort of precision soil solutions to support agriculture. And we're probably not going to be able to get rid of fertilizer completely, but I do think thinking about soil microbiome and soil supplements with bacteria, I think it should work. I think we should be working on this. But whatever, I'm biased. I'm preaching to myself. Yeah.
All right. Any other questions for Sean? So I have a question for you, going back to the fertilizer here for just a moment, and that is that as you pointed out, it's really critical. To what extent are the soil microbes sort of influenced by the temperature of the soil and things like that? And can you put some sort of discussion about how the changing climate is also impacting what's going on in the soil microbiology?
Yeah, I think it almost certainly is impacting the soil microbiology. There are folks who are thinking, deeply about this. I think temperatures of soils do impact the community profile of soils, right? And so I think as we think about, increased periods of temperature stress globally, I do think that that's going to sort of reshape the microbial community, and that's probably something that needs to be taken into account if we're trying to engineer soils or engineer microbial solutions, right? Temperature is a big thing, absolutely. Yeah.
One last question right here.
Oh, thanks. That was really nice.
Yeah.
Okay. All right, that's better. All right. The host-directed therapy is an interesting idea. That sounds great in your lab. We can block brucella and macrophage. Is that something that you think that pharmaceuticals are looking at in an antibiotic-resistant world? is that an approach that's going to have legs with the people who are going to really make these decisions in the pharmaceutical world?
Yeah, I think there's nonprofits who are funding this and thinking about this. I'm certain that there are host-directed therapies that the pharmaceutical world's working on. I know that there's definitely people who are also working on immune therapies for infectious disease, sort of modifying immune cells, right? Sort of like CAR T for infectious disease, right? There's people thinking about that too. We are really doing the fundamental work, and we get a lot of hits. We get FDA-approved drugs like loperamide, for example, right? That's Imodium, right? That works, right? But the pharmacokinetics and pharmacodynamics are not appropriate for treatment of brucellosis, and so it would require some downstream modification of those molecules. I think it's something that could work in concept, but how much pharma is investing in that concept right now, I don't know the answer to that.
All righty. Please join me in thanking Sean for a fantastic talk. All righty, we have come to the close. I hope you enjoyed today's classes without quizzes. I hope you learned something new and you got just a little glimpse into the really remarkable research and learning that both our faculty and our students are doing. And here's actually where you can help us out. Consider this your homework assignment, if you will. So in the not too distant future, we'll be sending you an email. And please take a time to fill out that email to let us know what you enjoyed most about today so that we continue to improve the event. You heard earlier about I guess it was the final exam is going to the dairy store. This is the only class where you get to do your homework after you do the final exam. But please take a time to fill it out just to let us know, provide us some feedback so that we can continue to improve. So as we come to the close of our Classes Without Quizzes event, I want to extend my heartfelt gratitude to each and every one of you who participated, as just showing up, as to give talks, all of you online, to support everything that we're doing. Today it's been a true celebration of the incredible research and discovery and innovation that's happening here in the College of Natural Sciences and goes on every single day. And lastly, I'd like to take a moment to recognize our Assistant Director of Alumni Relations, Melody Vinke. Melody, can you please wave? She's in the back. Let's give her a great hand. She's really done an amazing job, not just with today, but if you were with us for the awards ceremony last night, that as well. In fact, really this whole weekend she's been on, and so thank you. You've really done an amazing, amazing job. So as we conclude, I want to encourage all of you to continue to stay engaged with the College of Natural Science, whether through mentorship, collaboration, or philanthropic support. Your continued involvement really plays a vital role in the future of Michigan State University and supporting the success of future Spartans, especially those of us associated with the College of Natural Science. So thank you once again for joining us today. I look forward to seeing the positive impact that each of you will continue to make in the world. Safe travels, and of course, I cannot end an event without saying, go green.